Okay, well, welcome to the 18th CCHF Virtual Symposium on Frontiers of CH Functionalization. It's amazing. 2 minutes ago, there were only about 180 attendees at this meeting. And now we are up to 1 minute later, we're up to 520. And we know most people come in a little late anyhow. So it's interesting to see how many people are going to join today. Now, you know, we're all in a very difficult situation these days, not able to do any experiments and things like that, which is what we always like to do. So today we're going to begin the meeting with a couple of experiments. And so one of the things we want to do is increase the ability of people to be able to connect with this meeting. So the chat room and question and answers is fine, but we want to have some ability to do live engagement with the meeting. And so we're going to do some little experiments to test whether we can handle this more extensively at the next meeting. So I'm noticing that many of you have already gone to www.menti.com. And I would like everyone to go to their phones to sign up with that and put in this code 845526. Okay, great. So what I want to do first of all is to test this. And I can see already a lot of you are already testing it. Can't work in the lab, so they must have got up early this time around. And there's quite a lot of people coming in from Europe at 10 o'clock and 11 o'clock at night as well. So welcome to all of you. Okay, so next slide, please, Rio. Okay, so the reason why the CCHF did this and it did this these symposia, and it seems rather passé these days, but a few years ago, we had to learn how to communicate effectively to a large group in a virtual setting. And so once we established that, we really wanted to connect beyond the community of the CCHF into the whole community that are interested in CH functionalization. And back a few years ago, this was a major challenge. And of course, now we're all doing this every day in the classes using the Zoom type system. Although fortunately for us, we've been doing Zoom for nearly a couple of years now. So it's a very powerful way for us to communicate. So next slide, Rio. Now, it's interesting, our view of how to do these meetings has changed because our plan was actually to build communities. So we wanted everyone to join us from lecture halls and conference rooms from all over the world. And so these are some of the photos of those events. Everyone had to send us a photo to show the groups that they were with. And of course, we still want to build communities, but now we have to do it in a different way. We need to do it with everyone coming in individually, but hopefully in the future, we'll be able to go back to this community building where people join together to be involved in these meetings. Okay, next slide, please. Okay, so we've got a great lineup today. We've got 3 fantastic speakers, very exciting work, great presenters. So it's going to be a really exciting event. So we've got Alison Narayan beginning from the University of Michigan. Then we'll have Abigail Doyle from Princeton University. And then last but not least, we'll have Tom Rovis from Columbia University. And I'll tell you more about these people in a second, but for now, if we can go on to the next slide. Because I have to do a little bit of housekeeping with you. So the way we're set up here, everyone but the speakers, the microphones are muted. So if you want to ask questions, the thing you need to do is to go into the question and answers, which is down at the bottom of the Zoom page. And you can enter your questions at that time. And then I will be asking those questions on your behalf at the end of the talk. We used to insist that people would take a picture, of the group. So that's become a problem, okay? But we do want to have something else, because a lot of you are at home with pets around you. So the job these days is to take a photograph of you and your pet, because we would like to have a collage at the next meeting of all the pets that have joined this meeting with you. And then also, we'd like to have feedback from you about what you think of the event and how we do things. And this is, again, very easy to do. All you need to do is get your camera and point it towards this shape here, the QR code, and you'll get a link immediately that you can later on fill up and give us feedback. That's very valuable for us too, in terms of planning this meeting. For example, we've got Tom Rovis coming back for a second time because people thought he did so well when he spoke about 3 or 4 years ago, and his work has continued to grow. We actually have a series of meetings coming up these days. So next slide, please, Rio. We've actually teamed up with the ACS, the Division of Organic Chemistry. We actually technically run the meetings for them. And they have also expanded tremendously their programming as well. So we have 2 CCHF events coming up in the next few months. And then the ACS Division of Organic Chemistry has their own events as well. And do notice that the links for the meeting are different, and their time of day is different as well. But we encourage you to come to both of those symposia. Okay, so our next symposium after this one, it will be the 19th, and that is going to be quite an international event. We have Bob Knowles from Princeton, but we have Ang Lee coming in from Shanghai Institute of Organic Chemistry, a really fantastic total synthesis person who uses CH functionalization strategy in a really elegant way. And also Pedro Perez from the University of Leva in Spain. So it's a very international event. That's on Tuesday, June 30th. Next slide, please. We are also, as I said, the next one for the Division of Organic Chemistry is going to be on May 27th. But notice the time is different. That starts at 12 p.m. And so they have Teshi Gyun from Wisconsin, as well as an industrial speaker, Matt Bayo from Snapdragon Chemistry. Okay, next slide, please. And then our next center virtual meeting is going to be on September the 15th. And that will have Ken Hauk, a member of our center from UCLA, Masa Wasa, who's developing a really nice program at Boston College, and then David Proctor from the University of Manchester. So do look out for these schedules. We hope you will all join us. Okay, great. So next slide. Okay, so the reason why I spent a few minutes to talk is to allow the late audience to arrive. So we're now up to 820 participants, and that's just on Zoom. There's also a lot of people coming in on YouTube as well. So now we can start the real program. And it's a great honor for me to be able to introduce Alison Narayan, because Alison, we consider her as an alum from the CCHF. Because she actually did her PhD with Richmond Sarpong at the University of Berkeley, who is a faculty member of the center. And then she did a postdoc with David Sherman at the University of Michigan, another member of our center as well. And indeed, during her time with us, we saw Alison as the real role model on how to really do collaborative research across the center. She did a fantastic job. And not too surprisingly, she actually got hired by the University of Michigan in 2015, where she's an assistant professor of chemistry, as well as a research associate professor at the Life Science Institute that they have there as well. And indeed, her promise was rapidly recognized, because in 2016, she received the ACS Chem Society Chemical and Engineering News Talented 12. So recognizes some of the really exciting rising stars in the field of organic chemistry and broadly in chemistry. And indeed, that promise has really blossomed over the last few years. She's developed a really fantastic program. It's very exciting to see how it's developed, and we're delighted to have you here. And the title of the talk is Biocatalysis and Complex Molecule Synthesis. Welcome.
Hello, everyone. And Hugh, thanks for the kind introduction. Let me pull up my slide. So it says host disabled attendee screen sharing. Rio, help me out.
1 second, Allison. Sorry about that.
That's okay.
Don't worry, Allison. Rio is sweating at the moment.
I can keep going without the slides. That's okay.
Oh, don't worry, it'll be up in a second.
You should be good now.
Okay, let me try it again. Yeah, here we go. All right, can everyone see my slides? I got a thumbs up. I got a thumbs up from Hugh, so we're good to go. Well, it's really quite a treat to be participating in this virtual symposium today. Not only because I'm always excited to share the work from my research group, but specifically because—
Hi, Allison.
Yeah.
You're sharing the presenter's view currently. So.
Let's see.
So if you start that again.
Is that better?
That's better. Thank you.
All right. Thanks. So the CCHF community has been really important to me as an early career scientist. As Hugh pointed out, I had the opportunity to work with Richmond Sarpong during my PhD. And my first experience with the center was in the early days of my postdoc when I started in David Sherman's lab. And part of what really drew me into making this change between my PhD work and my postdoc was the idea that using biocatalysis and studying how nature builds molecules, you have different types of opportunity to achieve selective transformations. And the CCHF community was a really interesting backdrop for thinking about selectivity and reactions. On this slide, I have just a few molecules that were at the center of studies going on across the CCHF, looking at how we can challenge ourselves as chemists to achieve divergent selectivity on these molecules. And so it was really selectivity that lured me into working in biocatalysis. And I would say after spending several years working in this area, definitely enzymes can deliver with high levels of site selectivity, chemoselectivity, and also stereoselectivity. And I've also come to appreciate other elements of biocatalysis in synthesis, including the sustainability of these methods. Oops, oh no, sorry. Trying to get a pointer. There we go. Including the sustainability of using biocatalysis and synthesis and other advantages this offers, including the ability to streamline synthetic steps and have multiple catalysts compatible with each other in the single flask. And so based on these types of advantages, biocatalysis has been broadly applied by process chemists. These are just a few examples of pharmaceutical agents that have been synthesized using at least one biocatalytic step. And while most of these routes are based in traditional synthetic methods, weaving in biocatalysis for just 1 to 2 steps, a recent synthesis from Merck's process chemistry group, which was a collaboration with Codexis, I think fully illustrates the advantages of biocatalysis and synthesis, where here they envision not just introducing one biocatalytic step to make islatravir, but a total biocatalytic synthesis, taking advantage of the potential for multi-enzyme cascades in a single pot reaction. And so they envision stitching together this entire molecule using enzymes, and were able to engineer the enzymes appropriate for their specific substrates to ultimately achieve a sequenced one-pot synthesis of islatravir. And so I take a lot of inspiration from this synthesis, thinking about how do we do one pot total synthesis of complex molecules? And in my opinion, in order to enable more synthetic routes that are possible using this type of approach, there's a huge need for the development of additional types of biocatalytic reactions that chemists can reliably plan into their synthesis. And this is a huge motivation for my lab's work, where we're focused on discovering new enzymes, mostly from natural product biosynthetic pathways, and then understanding the full capabilities of these catalysts in synthesis, the breadth of their substrate scope, as well as other types of reactions that might be possible. And then engineering these types of reactions and planning them into synthetic routes to enable us to efficiently access molecules. And so I want to bring it back to this idea of selectivity, and specifically catalyst controlled selectivity, which can give you a way to efficiently build molecules. And so we think of catalyst controlled selectivity in biocatalysis having a few advantages when applied in synthesis. And so one thing that we're interested in within my group is the idea of using selective enzymes in order to do late stage modifications of molecules. And so here, this tricyclic molecule is the core that's conserved across the paralytic shellfish toxins, which are potent binders to sodium-gated, voltage-gated sodium ion channels. And we've been interested in studying how these molecules are assembled in nature because the proposal is that this tricyclic core can undergo a number of selective modifications to transform this into natural products like saxitoxin and higher oxidized versions of this natural product. And so this is a molecule that I first was inspired to think about based on my time in the center and thinking about controlled selectivity in CH functionalization reactions. And this is a project that one of the first graduate students in my lab brought to life. And so I won't focus on this project in my talk today, but I did want to give a plug for April's thesis defense, which is coming up this Friday. And so if you're interested in tuning into that, feel free to shoot me an email or contact me on Twitter, and I'm happy to give you the details so you can attend. All right, and so the other way that we think about catalyst-controlled selectivity in biocatalysis is thinking about how to assemble cores of molecules and reactions that typically challenge traditional methods for building complex molecules. And so one of the reactions we've been interested in is the idea that you can take phenolic compounds and do an oxidative dearomatization where this presents a challenge in both the site selectivity and stereoselectivity, but generates a reactive intermediate that could then be further elaborated in a cascade type process. We're also interested in thinking about how we can use biocatalysis in order to execute convergent syntheses. For example, taking two fragments and assembling those, controlling the site of bond formation. And so I'll start first with this oxidative dearomatization chemistry, where we were intrigued to work on this reaction, both based on the site selectivity and the stereoselectivity challenge presented by this transformation. Also thinking about fairly readily available starting materials, phenolic substrates that you might be able to buy or easily access in the lab can be transformed into building blocks where we've installed the stereo center and also generated a reactive dienone intermediate that can be primed for further chemistry. So this is a transformation that has been implicated in the biosynthesis of a number of different natural products. And chemists have also taken on this reaction where it's still an outstanding challenge to control the site selectivity in this reaction, where this is typically controlled by the substrate. But chemists have made advances in accessing and designing methods that have excellent stereo selectivity. And here the challenge is really crossing over into the regime where you can have a reaction that's catalytic in chiral information. And so we were interested to think about, can biocatalysis provide a way to do this reaction with controlled site and stereo selectivity? And might we be able to build a panel of enzymes with complementary selectivity to provide a tool for chemists to access and allow them to plan this reaction into their synthesis. All right, and so there is a class of enzymes that's capable of doing this transformation in nature, and they contain this flavin cofactor bound within the active site of the enzyme. Here, substrate binding triggers the reduction of that flavin cofactor to FADH2, which can then react with an equivalent of O2, here the stoichiometric oxidant in the reaction, in order to generate this hydroperoxyflavin, which can then engage with an electron-rich substrate such as a phenol to perform that oxidative dearomatization. So in this platform, the selectivity of the reaction is completely controlled with how the substrate is positioned relative to this hydroperoxyflavin cofactor. And so we thought that this might be a system where we could have catalyst-controlled selectivity. And when we're interested in starting this type of project, we of course need enzymes to start working with. And so one place that we go to find enzymes is from natural product biosynthetic pathways. Strategically, this is a great place to start because it can connect a set of genes to a given structure, where you can then have a blueprint for thinking about how that complex molecule was stitched together by a set of enzymes. We also think about going beyond enzymes that have been studied in the context of natural product biosynthesis more broadly in looking at the activity of enzymes across whole families, and we're also interested in fine-tuning the chemistry of these enzymes through established protein engineering protocols. So in the case of this oxidative dearomatization that we were interested in, there were a set of enzymes that had previously been identified to do this chemistry in the context of various natural product biosynthetic pathways. And so we chose these three enzymes, TROP-B, AZA-H, and SORB-C, as a starting point to explore this chemistry. And so once we had identified that these were the proteins that we wanted to work with, what we needed to do was go ahead and get those enzymes in our lab. And so the first step towards that is to buy a synthetic gene that encodes for the production of that molecule, which we can then use a heterologous host like E. coli and transform that organism with that DNA so it can produce our protein of interest. And then we can use that protein to run reactions and analyze them using standard techniques. So there are a few different ways that you can use a biocatalyst. You can take that culture of E. coli and put those cells directly into your reaction. This is really simple to do. We call this a whole cell reaction. You can also lyse open those cells using sonication or a chemical lysis step to generate lysate. This is basically the soup of the cell, and run reactions directly with that. Or you can take the time to purify your protein from others in your heterologous host, and then run what we call in vitro reactions. And so the first step in this project was to synthesize a panel of substrates that could test the reactivity and selectivity of this panel of enzymes. And so Summer Baker Dockery, upon joining the group, made a panel of about 50 different substrates and assayed their reactivity in analytical scale reactions. So this is a snapshot of that data where you can see that each enzyme, when we're looking at their activity across this whole panel, does react with a subset of these substrates, and that each enzyme has a unique fingerprint of the types of substrates that it will dearomatize. When we asked the next question about selectivity, we found that for the most part, enzymes tend to have a conserved site and stereoselectivity for what they do in their natural product biosynthetic pathway. So for example, in the case of TROP-B, this is the site of the reaction, and we always generate the stereocenter with the R configuration. The stereoselectivity of AZA-H wasn't known at the point we began this study. And ultimately we found that it installs that stereocenter with the same configuration as trope B, and uniquely would work on substrates that were less electron rich. And so from this point, we had a handful of enzymes that could successfully do this reaction across a panel of substrates. And we started to think about target natural products that we might be able to synthesize using this method. And one class of natural products we were very intrigued by based on their biological activities that had been reported are these azophylone natural products, which have this conserved bicyclic core. So there's been a lot of beautiful synthetic work done towards this family of natural products, including work from John Porco's lab, where they use an oxidative dearomatization strategy to access members of this class. As we began looking at various target molecules, we noticed that at this C7 position of the azophylone core, where we'd be installing this oxygen atom through the oxidative dearomatization reaction, we would need catalysts to give us either configuration on demand. But from the enzymes that we had studied initially, AZA-H and Trop B, we really only had access to one of these enantiomers. And so that caused us to think about strategically which way we wanted to go next, from working with these enzymes directly from biosynthetic pathways that had been studied. We considered taking a protein engineering approach. We also considered looking more broadly across the set of sequences. So this diagram is called a sequence similarity network, and it represents the sequences that are known in publicly available databases that are most related to these enzymes that we had studied. So from this data, we could start to think about sequences that are highly related to things like SORB-C, Trop-B, and AZA-H. And ultimately, we wanted to find a catalyst that could install this C3 stereocenter with the S configuration. And so we were able to identify an enzyme that did that as we profiled the activity of a whole series of these enzymes, and ultimately found that there were trends when we looked at the sequences of the enzymes that would give us the R configuration in this dearomatization versus the S. And so we obtained a crystal structure of Trop-B, one of those initial enzymes that we studied, and found that this tyrosine here was very important for substrate binding. We proposed that there's a hydrogen bonding interaction that helps to hold that substrate in place. What we found in the enzymes that give the enantiomeric products is that tyrosine is located on the other side of the pocket. And so we can confirm through mutagenesis studies where switching the location of this tyrosine can switch the outcome of the stereochemical outcome of the reaction. Okay, so we've used then these two catalysts in order to execute a chemoenzymatic stereodivergent synthesis of this class toward this class of natural products, where first we really needed to identify substrates that would work well with both enzymes. And so we were able to do that in identifying this enone substrate, which is processed in high efficiency by both AZA-H and AFOD. And so here we're able to carry out this dearomatization on preparative scale and then perform an acylation and canonical condensation in order to access this natural product, trichoflectin. And so one of the synthetic students, Josh Pizer in the lab, was working on this project and doing his due diligence and characterization, went to get the optical rotation, only to find that it didn't match what had been reported in the literature. So then he used AFOD to make the enantiomer of this building block and further elaborated that to trichoflectin with the R configuration. And after a series of CD studies and crystal structures, we were able to confirm that trichoflectin in the natural form is the R enantiomer. Okay, so we can apply this to a number of different natural products in this class. And we determined that not all of these have been misassigned. So for example, we can access lunativic acid, which was correctly reported in the literature, in order to demonstrate this strategy. And since then, Josh has gone on to make another number of other azophyllo natural products. So just in the last couple minutes, I wanted to tell you about a newer project going on in the lab. We're really thinking about how we can use biocatalysis to assemble molecules in a convergent fashion. So specifically, we were interested in biocatalysts that could potentially form biaryl bonds. So this is a common disconnection that chemists make when they're thinking about how to assemble molecules. And also because this motif is present in a number of very important molecules, both from a medicinal standpoint and also things that are important in asymmetric catalysis. And so if I asked you to do a retrosynthesis in how you might access something like a bienol derivative, you're likely to come up with one of the following strategies. So you can think about using a pre-functionalized approach where you can, for example, plan for a Suzuki coupling in order to construct that bond. Here, this can be challenging to do if you need to optimize your conditions for this reaction in order to get high levels of efficiency and selectivity for a given substrate. Also, the other approach that you could use is an oxidative coupling reaction. Here, the challenge is often controlling the site of bond formation, as well as the stereoselectivity with how these molecules can come together in space. So we're interested in thinking about how nature does this transformation. And there have been a handful of enzymes reported to do this type of oxidative coupling in the context of natural products. In fact, the month that I started at the University of Michigan, Michael Mueller's group reported a set of complementary catalysts that are able to form these types of biscoumarin products. And so we got the genes that encoded for these proteins and decided to test out this chemistry in our own hands. So we began to appreciate right away how challenging this reaction is to do when you're taking these more electron deficient phenolic compounds, coumarins, in attempting this type of oxidative coupling reaction. A lot of the standard conditions simply gave us no product. We were able to identify some conditions that can give us modest yields, giving a mixture of different site selectivities. And so we usually see what we call this 8,8' product is the major product. The second most abundant product, this 6,8' couple product. And then we would always see very small amounts of the 6,6' product under small molecule mediated conditions. And so what was really exciting is that one of these enzymes, KTNC, is perfectly selective for the 8,8' product, and DESC is mostly selective for the 6,8' product. And so we thought this might be a great platform to explore for catalyst-controlled selectivity in this oxidative coupling reaction. So some of our fundamental questions going into this project are, is the reaction scalable? Are these enzymes promiscuous in the substrates they'll operate on? Over a range of substrates, do we see maintained catalyst-controlled selectivity? And can we start to do things beyond dimerization and think about cross-coupling reactions? So for these reactions, we're running them in a fairly simple way. We're just doing straight up biotransformations where we're growing up yeast, producing each of these enzymes, tossing in our substrates, and then analyzing the reactions. And this works pretty well both on analytical scale and preparative scale. And so what we found when we began to look at the substrate scope, at least with a number of different coumarin substrates, we see that both enzymes seem to have a substrate scope where it tolerates various groups along the periphery of this coumarin. And we can also do this type of dimerization on other types of bicyclic phenolic compounds. One of the things that we saw that was interesting is that for the most part, the site selectivity of these catalysts is maintained across a number of different substrates. In the case of KTNC, we see with the native substrate, this is the major product. But as we start to increase the bulk of the group at this position from a methoxy to an ethoxy, we see a switch in site selectivity to that 6' product, which is the most difficult to access using small molecule methods. And if we continue to bulk that up, then we can exclusively access this 6' product. So from this point, our next challenge was thinking about if you can go away from dimerization to using these catalysts to do cross-coupling type reactions. And so Jessica Yazarians in the lab first tested out this concept. And here the challenge is that you're always fighting the native reaction, which is this dimerization. But she found by controlling the stoichiometry of the coupling partner, we're able to access cross-coupled products across to make a range of biscoumarin compounds. Further, she started to look at coupling partners where now we can cross-couple a coumarin with another type of phenolic substrate. And as you can see, we don't always get great yields, but we are able to observe these cross-coupled products. And this is really a great starting point for a biocatalysis project. It's really having that small amount of activity, which is key, because from there we can use established methods to improve the selectivity for those products, even if they're very minor. And so Laura Zecha in the lab has gone about designing a library of KTNC and DESC variants where now we can get various cross couplings with improved yields and tuning the site selectivity to favor specific products. So we think that although this is at an early stage, it's a really exciting result. And you could think about engineering these enzymes to make a range of different types of chiral ligands and also various natural products. Okay, and so at the end of at the point where this project stands now, we can start to answer some of these questions and we can say, yes, these reactions are scalable. They're somewhat substrate promiscuous. We can see catalyst controlled selectivity over a range of substrates, but there is also some influence based on the steric properties of the substrate, and that these reactions are not just limited to dimerizations. We can also start to see cross-coupling reactions. Okay, and so that's the sum of the chemistry that I wanted to share with you today that's focused on this catalyst-controlled selectivity in biocatalytic reactions. Here's the group. I acknowledged most of the students that contributed directly to these projects along the way. I didn't have Laura and Jessica's pictures. They worked on this cross-coupling chemistry. And finally, I just wanted to put in a plug, since this is the virtual audience, for a virtual biocatalysis meetup if you're interested in learning more about biocatalysis. So we started this a few weeks ago with scientists from Merck and the University of Manchester, and it's really grown to include some pretty famous faces. So we decided that we would keep this up. So if you're interested, you can go to this Google form and get added to the mailing list, or you can contact me directly and I can add you to the list so you can get more information. And if there's time, I'd be happy to take any questions.
I'm the only one who's clapping, so but great talk.
Thanks, you.
So we do have a few questions that have come in. So if you don't mind, I will read them to you. We got a question from Augustin Gus Pinot, who says, outstanding talk with an exclamation mark and beautiful biocatalysis. A practical question, how difficult is it to extract the targeted molecule from the reaction mixture?
That's a great question. So like any chemistry that you're working on, the answer really depends on the properties of that molecule. So some things are tougher to work with than others. And so here we're always thinking about how do you extract something out of water. And so the students in the lab have gotten pretty good at getting the right blends of solvents to do extractions and things like that. In worst case scenarios, when we have very water soluble molecules, we might need to do a derivatization step to enable the isolation.
Okay, thank you. And there's actually a huge collection of questions, so I'll only be able to ask you a couple of them, and hopefully you can go to the question and answer afterwards to answer them yourself. But next question is from Josiah Newton. Why was the optical rotation of the two enantiomers so different? Do the enzyme yield mixtures of enantiomers?
So in the azophylones, yeah, so the magnitude of those samples was different with the different signs. And so that's I think more a question about the polarimeter that we're using in the scale that we were making that measurement on, and not a product of the purity of the compounds. And so we did do an SFC analysis, a chiral separation by SFC, to show clearly how much of each enantiomer we have in those reactions. So usually these reactions we're getting greater than a 99% EE.
Okay, great. And then one final question out live. Have you confirmed, this is from Bram Frohock, have you confirmed the biological activity of the first set of natural products made using the oxidative dearomatization?
That's a really great question. And so that's ongoing work. And so I think it's really important to learn about the molecules that we make and not just put them in a freezer for them to stay there forever. And so we do a few different things with our molecules. The first thing that we do is we can put them into the screening collection that we have at the University of Michigan. And so we can passively learn about our compounds that way from others that are doing biological screens. But more recently, Josh Pizer, who is working on that project, has coupled with Johnny Sexton. His lab develops high content, high throughput biological assays, and specifically looking at cell painting assays, where we can get a lot of information about the parts of the cell our compounds are affecting. And so that has generated some exciting data.
Okay, great. Well, Alison, thanks very much for a wonderful talk. Unfortunately, this is not the time for you to rest because I'm seeing 17 questions for you in the questions and answers. So I think you'll have a good discussion going further forward as well.
All right, thanks you.
Thanks very
much. I'm trying to stop share, but it won't let me select. Let's see. There, thanks.
Okay, excellent. So while we're waiting to get ready for the next talk, I thought you'd be interested to know we have over 1,100 people joining us at this stage, about 872 on Zoom and over 230 coming in on YouTube. So thank you all for joining us, and looking forward to the rest of the session. Okay, so our next speaker is Abigail Doyle, who's from Princeton University. So Abigail got a bachelor's and PhD at Harvard University with Eric Jacobson, and then began as an assistant professor at Princeton in 2008, but quickly made an incredibly big impact in the field because by 2017, she was the Barton Hepburn Professor of Chemistry, the chaired professorship in a very short period of time. In her early work, she made some really important developments in nickel catalyzed chemistry, and then combined it with photoredox chemistry that opened up a lot of really interesting opportunities in synthesis in general, carrying out fast reactions, and also extending it towards CH functionalization applications. And she has been recognized for her work. Earlier in her career in 2013, she got a Cope Scholar Award from the ACS. And in 2019, she got the 15th Hirata Award, a very prestigious award for her work as well. So Abigail, it's great to have you here, and we're very much looking forward to your talk. The title of your talk is Photocatalysis with Nickel.
Thank you, Hugh. Can everyone hear me?
Yes, we can hear you really well.
All right, and slides are up. Okay, well, thank you very much for the invitation. I think now more than ever, I'm grateful for the opportunity, right, to connect with our community in this manner and to share chemistry. I'm sure many people feel similarly So thank you to the CCHF for hosting these virtual symposia. About 5 years ago now, my group became interested in combining visible light photoredox catalysis with nickel catalysis to enable cross-coupling reactions that weren't otherwise possible with nickel, and for that matter, palladium catalysis alone. And through the contributions of many other research groups, this is an area that has developed and I think has become particularly enabling for coupling abundant and stable molecules such as carboxylic acids, amine derivatives, ether derivatives as surrogates for organometallic reagents in cross-coupling. The role of the photoredox catalyst in most of these cases is to undergo photoinduced electron transfer with a substrate to generate a substrate radical, or to undergo photoinduced electron transfer with a nickel catalyst to modulate its oxidation state. And given the impact that this single sort of elementary step has had on nickel catalyzed cross-coupling, my group became interested in learning about other mechanisms that where in transition metals harness visible light energy to, and whether any of these alternative mechanisms could be deployed in nickel catalyzed cross-coupling to design sort of new reactions. And the specific elementary step that caught our attention was photoinduced ligand dissociation. This is an elementary step that actually has an important history in sort of early examples of CH activation. But the specific example that we were interested in is from a study that the Nocera laboratory published in 2015, where they showed that these nickel-3 trichloride complexes can absorb visible light, and in doing so, undergo a photoelimination of chlorine radical. So this caught our attention because chlorine radical is a potent hydrogen atom abstractor, and in fact can abstract some of the strongest sp3ch bonds in free radical chlorinations. However, chlorine radical is typically generated from highly oxidizing chlorine sources, like chlorine gas, usually in high concentrations and using UV light. And so we envisioned that this photoelimination approach could address each of these limitations, and in combination with nickel cross-coupling, enable sp3ch cross-coupling of stable or sort of abundant alkanes. In particular, the way that we thought that this might be affected is that we could use an organic chloride like an aryl chloride as both a cross-coupling partner and a source of chlorine radical, that it would undergo oxidative addition to nickel 0 to generate a nickel 2 complex. We envisioned using a photoredox catalyst to oxidize nickel 2 to nickel 3 through photoinduced electron transfer. And then based off of the precedent from the Nocera lab, we proposed that this nickel 3 complex could undergo photoelimination to generate chlorine radical. Hydrogen atom abstraction with a substrate bond would generate an organic radical that could recombine with nickel, and then reductive elimination would generate our CH cross-coupled product. And finally, single electron transfer would turn over both the nickel and the iridium catalyst. So to test this proposal, Ben Shields, who's a graduate student in my lab, decided to explore application of this strategy to the alpha arylation of THF. And what he found through experimentation is that this fluorinated iridium photocatalyst, in combination with a nickel catalyst and di-2-butyl bipyridine as ligand, could in fact enable the alpha arylation of THF as well as other ethers. And so this is a select scope of this reaction, but I'm not going to go into it in any detail here. What I will point out is that at the same time that we sort of published this work, there were 2 other methods that were published that are nickel photoredox methods to accomplish similar, albeit mechanistically distinct transformations from the Molander lab and the Macmillan lab. One interesting difference between our work and theirs is our use of aryl chlorides, and in fact our reliance on aryl chlorides since it's supplying the chlorine radical for hydrogen atom abstraction. And interestingly, in their work, so they use aryl bromides and iodides, and interestingly, in the vast majority of nickel photoredox coupling methods do use aryl bromides and iodides as coupling reactions. A student of mine just recently did sort of a quick survey of this and found of the 450 papers or so that have been published on nickel floater redox catalysis, only 10 of them about use aryl chlorides as coupling partners compared with the bromides and iodides. And most of those come, in fact, from this photoelimination strategy that I'm going to tell you about today. And so in thinking about applications of this approach in synthesis, we became sort of focused on sort of considering applications that would sort of take advantage of the use of aryl chlorides, in particular their abundance and low cost as compared to aryl bromides and iodides. And so the first application that came to mind, and this is work from Matt Nielsen, Ben, and Janie, was to consider preparing benzaldehydes from aryl chlorides through selective CH functionalization of dioxylane. And this was appealing to us because benzaldehydes are, of course, very versatile intermediates in synthesis, yet are still used primarily at early stages in synthesis due to some of the limitations associated with their preparation. In particular, things like Vilsmeier-Hack chemistry or aryl anion chemistry limit functional group tolerance. We also noticed that there are orders of magnitude fewer commercially available benzaldehydes compared with aryl chlorides. And so a goal was to demonstrate that the CH functionalization, sort of our nickel photoredox strategy, could enable access to both early stage